Imagine this for a second. You've got this tiny, single-celled organism, Didinium, and it's about to do something, well, kind of mind-blowing.
Oh, yeah.
It's going to completely swallow another organism, Paramecium, that's the exact same size as itself.
Wow. Okay, same size. That's impressive.
Right, it's like a person trying to swallow a whole month's worth of groceries in one go. It sounds impossible. And the earliest life forms, prokaryotes, they just can't pull off anything like that.
No, they really can't. And that's the crux of it, isn't it?
Yeah.
That ability, that dramatic shape changing, it signals a fundamental leap in complexity compared to the simpler prokaryotes.
Absolutely. That maneuverability is pure eukaryote. So today we're doing a deep dive into exactly that. The origin and diversification of eukaryotes, straight from your Campbell Biology and Focus textbook.
Right. Our mission here is really to trace that amazing journey. How did these incredibly complex, shape-shifting eukaryotic cells actually come about from their much simpler ancestors? We want to paint a picture you can follow without needing diagrams.
And we're going further, looking at how multicellular life got started, mapping out the big branches of the eukaryotic family tree, and importantly, exploring the huge impact these tiny organisms have on ecosystems, even on our own health.
Yeah, there are some real surprises in store. This is where life on Earth gets incredibly diverse and, frankly, pretty interesting. Get ready for some aha moments.
Okay, so let's start unpacking. What really sets a eukaryote apart from those early prokaryotes?
Well, for billions of years, prokaryotes had the planet to themselves. They're survivors, absolutely, but structurally quite simple. Eukaryotes brought major changes. They have a nucleus, a dedicated spot for their DNA and other membrane-bound compartments organelles, right?
Like mitochondria, the Golgi.
Exactly. Mitochondria for energy, Golgi for processing proteins. Each organelle has a specific job. It makes the cell way more organized, more efficient than a prokaryote, which is more like an open-plan studio apartment.
Huh. I like that. And this internal organization, especially the cell's framework, that's key to the shape-shifting we mentioned.
Precisely. The cytoskeleton is crucial. Think of it like internal scaffolding, but made of proteins. And it's incredibly dynamic, constantly building, breaking down.
So it's not rigid.
Not at all. It gives support, sure, but it also lets eukaryotes have these irregular shapes and, importantly, change shape dramatically for eating, like didinium or moving or growing. Prokaryotes, most have rigid cell walls and a much simpler cytoskeleton. They just don't have that kind of flexibility. It's like comparing, I don't know, a tent frame to a building steel structure.
That makes sense. So this new level of complexity, when did it actually appear on the scene? Let's talk deep time, the fossil record.
We're talking vast timescales. Prokaryotes show up around 3.5 billion years ago,
Okay.
but the oldest widely accepted eukaryotic fossils, they're much younger, around 1.8 billion years old.
1.8 billion. It's still incredibly old, but a big gap after prokaryotes.
It is. Although there's chemical evidence, specific types of molecules in ancient rocks that hints eukaryotes might have been around even earlier, maybe 2.7 billion years ago. But physical fossils are trickier to find and confirm from that far back.
Okay, so after prokaryotes ruled for so long, what does the fossil record tell us about how eukaryotes started to branch out to diversify?
We see it happening in roughly 3 stages, based on the fossils. First, what we call the initial diversification. That's from about 1.8 down to 1.3 billion years ago.
What did it look like?
Mostly single-celled eukaryotes, but with some variety in size and shape: spheres, ovals, spindle shapes. We also see the very first hints of simple multicellularity, like basic filaments of cells.
Okay, basic forms. Then things got more complex.
Definitely. The second stage, the origin of novel features, runs from about 1.3 billion years ago up to 635 million years ago. This is where things get really interesting. We see the evolution of complex, multicellularity organisms with different cell types doing different jobs.
Like specialized tissues.
Sort of the precursors, yes. And sexual life cycles appear, plus widespread photosynthesis within eukaryotes. We have great fossils like Bangiomorpha, a red alga from 1.2 billion years ago that clearly shows differentiated cells. It looks remarkably like modern red algae.
So for billions of years, life was tiny, then suddenly bigger things appear.
Pretty much. The third stage is the emergence of large eukaryotes from 635 to 535 million years ago. Before this, life was almost entirely microscopic. Then, boom, the Ediacaran biota.
Ah, yes, the Ediacaran, weird, soft-bodied things.
Exactly. Some were over a meter long, Soft-bodied, yes, often quilt-like or fern-like in structure. It was a dramatic increase in size, in diversity, in just the sheer variety of shapes. This really set the table for what came next, the Cambrian explosion.
It sounds like eukaryotes are kind of cobbled together from bits of other organisms. You mentioned they have genes from both archaea and bacteria.
That's actually a pretty good way to think about it. Molecular data shows eukaryotes are combination organisms. Some genes, some cellular processes look archaeal, others look bacterial. This genetic mixing is explained by the endosymbiont theory.
Okay, endosymbiosis. Let's break that down. What's the core idea?
The theory proposes that mitochondria, the powerhouses of the cell and plastids, which include chloroplasts for photosynthesis, were originally free-living bacteria.
Living on their own?
Yep. And at some point, they were engulfed by a larger host cell, maybe an early archaeal cell, and instead of being digested, they started living inside. A permanent partnership formed.
Like cellular tenants that became essential parts of the house.
Exactly. Over time, they became indispensable. It's a fantastic example of evolution driven by cooperation, or at least by a relationship that benefited both parties eventually.
So which came first in this partnership? The mitochondria or the plastids?
The evidence strongly points to mitochondria first. It seems the defining moment for all eukaryotes was when an ancestral host cell, likely an archaeon with some existing complexity like a flexible membrane and basic cytoskeleton, engulfed an aerobic bacterium.
An oxygen-using bacteria.
Right. And this partnership, this endosymbiosis, seems to have happened just once. It gave rise to the common ancestor of all eukaryotes that have mitochondria today, or at least remnants of them.
One single event that changed everything. What makes scientists so sure this actually happened? The evidence must be pretty good.
Oh, it's compelling. Really compelling. First off, the inner membranes of mitochondria and plastids. They have enzymes and transport systems very similar to those found in the plasma membranes of living bacteria today.
Okay, membrane similarities. What else?
They replicate inside the cell by a splitting process that looks a lot like bacterial division. Plus, they have their own DNA.
Their own DNA, separate from the nucleus.
Absolutely. And it's usually a circular molecule, just like bacterial chromosomes, and it lacks the histone proteins typically associated with eukaryotic DNA in the nucleus.
Wow, okay.
And there's more. They have their own ribosomes, the machinery to make proteins from their DNA instructions. And crucially, these ribosomes are more similarly in structure to bacterial ribosomes than to the eukaryotic ribosomes out in the cell cytoplasm.
So they really look and act like internal bacteria in many ways.
They do. And genetic sequencing has even pinpointed the likely ancestor of mitochondria, a group called the alpha proteobacteria.
That's pretty definitive. So mitochondria came first. What about the plastids, the photosynthetic ones?
They came later, much later. The idea is that a lineage of eukaryotes that already had mitochondria, so they were heterotrophic, eating things, then engulfed a photosynthetic cyanobacterium.
A blue-green alga, basically.
Right. And that event, called primary endosymbiosis, gave rise to the lineages that include red algae and green algae, and by extension, land plants, which evolved from green algae.
Okay, primary endosymbiosis gave us the main photosynthetic lineages, but you hear about secondary endosymbiosis too. Does it get even more complicated?
Oh, it does. It's like Russian nesting dolls sometimes. On several separate occasions later in evolution, a eukaryotic cell actually engulfed another eukaryotic cell that already contained plastids specifically, a red or green alga.
Whoa, okay. A eukaryote eating another eukaryote and keeping its chloroplasts.
This essentially, yes. That's secondary endosymbiosis. And we see the evidence. For example, in some groups, the plastids have more than 2 membranes around them, suggesting layers from the engulfing process.
Any really striking examples?
One of the most amazing is a group called the chlorarachniophytes. They engulfed a green alga. And get this, inside their cells, you can still find a tiny remnant nucleus from that engulfed green alga.
A nucleus within a nucleus, almost.
Well, a tiny vestigial one, yeah. It's called a nucleomorph. It even has a few functional genes left that are clearly related to green algae. It's like a living fossil record inside the cell, direct proof of that secondary engulfment. It's incredible.
That really is mind-blowing. Okay, so eukaryotes get complexity. They get organelles through endosymbiosis. What was the next huge leap?
That would have to be multicellularity. Having that internal complexity, that cytoskeleton, that ability to specialize, it opened the door for cells to cooperate and form larger structures.
Going beyond just single-celled life, how did that start? Was it one single breakthrough that led to animals, plants, fungi?
Not at all, and that's maybe one of the biggest takeaways here. The first steps were likely simple colonies, just cells sticking together after dividing, maybe with a little specialization. We still see those today.
Like algal filaments.
Exactly. But complex multicellularity, where you have significantly different cell types forming tissues and organs
Yeah.
that evolved independently multiple times.
Multiple times, really.
Yes. Think about it. Red algae, green algae, leading to plants,
Hmm.
brown algae, fungi, and animals. All of these groups achieved complex multicellularity, but they evolved it separately from different single-celled ancestors. It's a stunning example of convergent evolution at a grand scale.
Okay, that is a big deal. Multiple independent inventions of being complex and multicellular. Do we have examples of how this might have happened? Maybe looking at the genetics.
We do. A great model system is the Volvox lineage of green algae. You can find living species that show a clear progression. You start with single-celled Chlamydomonas, then you see simple colonies where cells are pretty much identical, and finally you get to Volvox itself.
And Volvox is different.
Oh yes, Volvox is a hollow sphere of cells, but it has two distinct specialized cell types: smaller somatic cells that handle movement and photosynthesis, and larger reproductive cells. It's true multicellularity with division of labor.
So how did it achieve that? Did it need a whole bunch of new genes?
That's the fascinating part. Apparently not.
Mhm.
Studies suggest the transition to multicellularity in Volvox, and likely in other groups too, involved mostly the repurposing of existing genes. Genes that had one function in the single-celled ancestor were sort of co-opted or modified to play new roles in cell communication, adhesion, or differentiation in the multicellular form.
So evolution tinkering with the existing toolkit rather than inventing brand new tools from scratch.
Precisely. It's about using old genes in new ways or new combinations.
And does this idea of gene co-option hold up when we look at the origin of animals?
It really seems to. The closest living single-celled relatives to animals are thought to be the choanoflagellates.
Choanoflagellates.
Yeah, the tiny protists, some are colonial. Morphologically, their collar cells used for feeding look almost identical to certain cells in sponges, which are very basic animals.
Okay, a structural similarity. What about genes?
Genetically, it's even more compelling. Choanoflagellates have genes for many proteins that were once thought to be unique to animals, especially proteins involved in making cells stick together, adhesion, and communicate, signaling. Things like cadherins.
Cadherins are key for animal tissues, right?
Absolutely. And it turns out that animal cadherins seem to be built using protein domains already present in choanoflagellates, with just a novel bit added. So again, it looks like the animal lineage repurposed these existing tools for building multicellular bodies.
Wow. Okay, so we've got complex cells, endosymbiosis, multiple origins of multicellularity. Let's try to get a handle on the sheer diversity. You mentioned the old kingdom protista is gone. Why was that necessary?
It was basically a taxonomic junk drawer. Anything eukaryotic that wasn't clearly an animal, a plant, or a fungus got dumped into protista. But molecular data, DNA sequencing mainly, showed it was massively polyphyletic.
Meaning they didn't all share a recent common ancestor.
Exactly. Some protistas were actually more closely related to animals than to other protists. Others were closer to plants or fungi. The grouping just didn't reflect evolutionary history at all, so it had to be dismantled.
So if not protista, how do biologists organize eukaryotes now? What's the current thinking?
The current model, though it's still debated and refined, organizes most eukaryotes into four large supergroups. The idea is that these four major lineages possibly diverge from each other relatively simultaneously. Finding the exact root of the whole eukaryotic tree is still tricky.
4 supergroups. Can we quickly run through them? What defines them?
Okay, first up is excavata. Many of these have a distinctive scooped out or excavated feeding groove on their side. It includes some notorious parasites like Giardia, causes hikers diarrhea.
Right, from contaminated water.
Yeah, and Trichomonas and STD, also Trypanosoma, which causes African sleeping sickness. Many excavates have really weirdly modified mitochondria or lack them entirely, often living anaerobically.
Okay, excavata. What's next?
Then there's the massive SAR supergroup. The name is an acronym for the three major clades within it: straminopiles, alveolates, and rhizarians. This group is incredibly diverse and likely arose from a secondary endosymbiosis event involving a red alga.
Wow, okay. Examples from SOAR.
Oh, tons.
Thanks.
Stromatophiles include the diatom's vital photosynthetic algae with beautiful silica shells like glass boxes. They're hugely important for aquatic food webs and even CO2 levels. Also, the brown algae, like giant kelp forests, they're the largest, most complex algae.
Wicked forest, okay. And alveolates.
Alveolates have little membrane-bound sacs, or alveoli, just under their cell surface. This includes dinoflagellates, some cause red tides, some are bioluminescent, they have cellulose plates, and ciliates, like paramecium, covered in cilia from moving and feeding.
And the R in SAR.
Fusarians. These are mostly amoeba-like organisms, but typically with thin, thread-like pseudopods, different from other amoebas. Includes forams, which make shells of calcium carbonate. Their fossils form chalk and limestone. And cercozoans are a really diverse group, some even photosynthetic through, yet another, independent endosymbiosis.
Goodness. Okay, 2 supergroups down. What's the third?
Archaeplastida. This is the lineage that directly resulted from that primary endosymbiosis event we talked about, the one where a eukaryote engulfed a cyanobacterium.
Yeah.
So this group includes red algae, green algae, and crucially all land plants.
Ah, so plants are nested within the supergroup.
Exactly. Red algae get their color from a pigment called phycoerythrin, letting them live deep underwater. Many are seaweeds, like nori for sushi. Green algae are the closest relatives to land plants, sharing very similar chloroplasts. They range from single cells like chlamydomonas to multicellular forms like sea lettuce, ulva, and volvox.
Right. And the final supergroup?
Includes us, right?
Yes. The fourth is Uniconta, another incredibly diverse group. It includes animals, fungi, and also some protists that are closely related to either animals or fungi. It's split into 2 main branches,
Achar.
the amoebozoans and the okistoconts. Amoebozoans are the amoebas with lobe or tube-shaped pseudopods, like the classic amoeba proteus you might see in labs. It also includes the fascinating slime molds.
Slime molds, they're the ones that can act like single cells but then come together?
Some do, yeah. Cellular slime molds like Dictyostelium live as solitary cells, but when food runs out, they aggregate into a multicellular slug that crawls around, eventually forming a stalk to release spores. It's another independent evolution of cooperative multicellular behavior. Amazing. And the other branch of Uniconta.
The opisthocons. This includes fungi, animals, and their closest single-celled relatives, like the nuclearides, related to fungi, and those choanoflagellates we mentioned, related to animals. This really drives home why Protista didn't work. Animals and fungi are embedded deep within these eukaryotic lineages, closely related to certain single-celled groups.
It's just incredible this microscopic world gave rise to everything we see. But let's bring it back to the protists themselves, the ones that aren't animals, plants, or fungi. What's their impact on the world today?
Immense. Even though many are single-celled, they are staggeringly complex internally. Each cell has to do everything: feeding, respiration, reproduction, response, using its organelles, like tiny organs. And ecologically, they are powerhouses.
You mentioned photosynthetic protists are major producers.
Huge. They form the base of most aquatic food webs. Globally, photosynthetic protists, along with cyanobacteria, carry out something like 30% of all photosynthesis on the planet. Think about that nearly a third of the oxygen we breathe and the food generated comes from these often microscopic organisms.
Wow. Diatoms, dinoflagellates.
Exactly. Diatoms especially are critical for the carbon cycle. When they die, their silica shells help them sink rapidly, carrying carbon down to the ocean floor. It's a major way CO2 gets locked away from the atmosphere long term. The biological carbon pump.
But that sounds vulnerable. What about climate change?
It's a serious concern. Rising sea surface temperatures can stratify the water column, meaning warmer surface layers don't mix well with colder, nutrient-rich, deeper waters.
So the protists at the surface run out of food.
Essentially, yes. Less nutrient upwelling means less growth for these photosynthetic protists. This can impact fisheries, marine ecosystems, and that vital carbon pump we rely on. It's a major feedback loop researchers are watching closely.
Okay, so huge role as producers. What about other interactions? Symbiosis?
Absolutely. Protists are involved in all kinds of symbiotic relationships. Many are crucial mutualists. The classic example is dinoflagellates living inside coral polyps.
The zooxanthellae.
Right. They photosynthesize and feed the coral, and in return get a protected environment and nutrients. Coral reefs literally depend on this partnership. Or think about the protists living in termite guts. They're the ones that actually digest the cellulose in wood, allowing the termite to get nutrition.
Essential partnerships. But there's a dark side too, right? Parasites.
Definitely. Protists include some devastating parasites. Phytophthora ramorum, causes sudden oak death, wiping out oak trees, and historically, Phytophthora infestans, the cause of potato late blight.
The Irish potato famine.
Exactly. Led to immense suffering and emigration, and it's still a major threat to potato and tomato crops today. These aren't trivial infections. They can reshape ecosystems in human societies.
And finally, the direct impact on human health itself.
Yes, some protists are major human pathogens. We mentioned trypanosoma, causing sleeping sickness. Its strategy for evading our immune system is incredible. It has thousands of genes for different surface proteins and constantly switches which one it displays.
Like changing its coat constantly so the immune system never gets a lock.
Precisely. It's called antigenic variation, a brilliant and deadly defense mechanism. About a third of its entire genome is dedicated just to this protein switching.
That's dedication to evasion, and the really big one globally, malaria.
Malaria, caused by Plasmodium, another protist, and apicomplexan. It has a complex life cycle, shuttling between mosquitoes and humans. It also uses antigenic variation and hides inside our red blood cells and liver cells to evade immune attack.
And finding a vaccine is notoriously difficult.
Extremely difficult, partly because of that variation and its life cycle complexity. But researchers are making progress, targeting different stages or aspects of its biology. One interesting target is the apicoplast, a weird, non-photosynthetic plastid that Plasmodium has, likely from secondary endosymbiosis way back. Because it has metabolic pathways different from ours, it's a potential target for drugs that would harm the parasite, but not us.
Fascinating. Okay, wow. We've really journeyed far today. Started with Didinium gulping down paramecium.
Which really set the stage, showing the unique capabilities unlocked by eukaryotic structure, born from endosymbiosis, these amazing combination organisms.
We traced their history in the fossil record, from early single cells to the first large complex life forms. And we really dug into that endosymbiont theory, mitochondria first, then plastids, even secondary events creating more complexity.
Yeah, and the realization that multicellularity wasn't a one-time trick. It evolved independently in animals, fungi, plants, and different algae groups, often by cleverly repurposing the genetic toolkit they already had.
Then we tried to map that incredible diversity using the 4 supergroups, Excavata, SAR, Archaeplastida, and Uniconta, getting a glimpse of the sheer variety of eukaryotic life.
And finally, we saw just how critical these organisms are, as the base of food webs, as climate regulators, as partners in symbiosis, and unfortunately, as causes of major diseases. They might be small, but their impact is enormous.
It really underscores how fundamental these evolutionary steps were, shaping the entire biosphere.
Absolutely. It all builds on these foundational events.
So, to leave you with something to think about, isn't it kind of profound that one, maybe 2 ancient engulfing events, one cell swallowing another, ultimately paved the way for this explosive diversity? For everything from an amoeba shuffling in pond water to, well, us. What does that tell you about how seemingly small biological innovations can completely change the trajectory of life on a planet?